We'll move on to Khuloud Jamal from King's College in London, and she'll also be talking about brain delivery and multimodal imaging of this process. Khuloud, please.
So thanks, Tuan, for the invitation, and Piat as well. So it's quite challenging to speak after three impressive speakers, actually, but I will try. So today I'm going to talk to you about multimodal imaging of nanotubes for crossing the blood-brain barrier. So I will apologize from Tuan because that's not purely Theranostics, because there's no particular therapeutics here. But I think before we go to this level, we have a lot of questions to answer on the delivery aspect, which employs different imaging modality techniques. So nanotubes, the structure I'm going to talk to you about today is this one. So it's one of the different carbon forms available. And we're interested in this because it has a different shape. So it has a long aspect ratio, which makes it different from the rest of the spherical nanoparticles. So one of the main limitations here is the service properties of the material. So if you can imagine a single nanotube is practically one rolled up layer of graphene material, but you can see there are a lot of aromatic rings here, which makes it actually water insoluble, and it's practically insoluble in many organic solvents. So a single walled nanotube is made of one layer of graphene, but multi-walled nanotubes are made of several layers, but they share the property that they are practically insoluble. So the first thing we need to perform is service functionalization of the material, and that makes it more water dispersible and improves the biocompatibility of the material. So in 2011, we published this study showing in 3D manner the direct crossing of nanotubes into a cell membrane. And this is quite important. Although there is no direct correlation between efficacy and method of delivery, but actually it gives us the opportunity that there are multiple ways of internalization into the cell, which could have multiple targets inside the cell, if so. So we performed this study to show if actually a CNT mediated delivery could improve the status of, in this case, we used rodents. And the idea was to inject a therapeutic siRNA in combination with the CNTs after intracerebral administration, just to see if that delivery could actually lead to internalization of the nucleic acid inside neurons and have any improvement. And what we could see is actually there was promising results where mice injected with the siRNA and the CNTs showed performance in their therapeutic behavior after subjection to ischemic stroke. And you can see the CNTs are widely distributed in many types of cells, including neurons and astrocytes. So because intracerebral administration is not really of relevance for patients, we would want to attempt the intravenous administration. And in this study, we did a direct comparison between what we called it by then wide and narrow CNTs with particular diameters. And interestingly, what we could see is you have brain uptake, which was more enhanced with the narrow CNTs, but in addition, you also have uptake in other types of organs. And the uptake was approximately 2 to 5% injected dose per gram tissue, which is quite substantial. So here we rely on radio labeling to track the CNTs. So this is Spec CT image, shows exactly where they go in the brain. And you can see there is a wide distribution across the whole brain, and it's quite quick. It happens within half an hour, sustained till 4 hours, and then it completely clears after 24 hours. So the first thing we would think, are we tracking the CNTs or the radio label? But if you look at the India DTA on its own, it's actually in the blood vessels and everywhere, but not the brain. So it's definitely something to do with the CNTs crossing the blood-brain barrier, perhaps, and reaching the CNS. So that opened our eyes on interaction with the blood-brain barrier, and we want to answer the question is, do CNTs cross the blood-brain barrier? If so, how? And going from actually a cell-level imaging to a whole brain imaging. So it's quite challenging because you can see the blood-brain barrier represented by the neurovasculature unit has many layers. So you have the endothelial cell, which is just a single layer with prominent tight junctions that actually makes the crossing of many molecules quite challenging. You have association with other types of cells, with the endothelial cells. So that kind of association causes the polarization and formation of tight junctions, which allows very limited amount of specific properties. So I'm not going to talk much about this, but actually you can see that there are different routes for crossing. So you have the water-insoluble material versus the soluble material. You have specific transporters and receptors for amino acids or important proteins that the brain needs. And very recently, there is a mechanism explored looking at the adsorptive mediated transcytosis for cationic nanoparticles. So what we want to explore is actually one of these mechanisms to enhance the crossing into the brain. So last year we had the first report on CNTs crossing the blood-brain barrier, and here we use primary brain endothelial cells, which is characteristic of formation of tight junction. So it's a truly representative model, co-cultured with astrocytes, and what we have shown is actually the CNTs are able to transcytose across the brain endothelial cell. It's a slow process, happens over 48 hours, and it's actually very specific. It's energy dependent, and it could be stopped if you deplete the energy from the system. You can also target cells beyond the blood-brain barrier, and in this case, the astrocytes. So we utilize the concept of double targeting here, and double targeting relies on a receptor that's overexpressed on the blood-brain barrier, but also on a target beyond, and in this case, a glioma. So we use a particular peptide here called angiopep2, which recognizes LRP1 receptor, which is as I mentioned, expressed on the brain capillaries as well as the glioma. And it has been already shown that this mechanism of active targeting can be coupled to other types of nanoparticles and improves the survival of animals. So why not to combine it with the CNTs? So we always rely on having a chelator here to allow for nuclear imaging, and then we prepared what we call non-targeted or targeted CNTs. There are various methods to quantify how much of the angiopep is conjugated. So, for example, this is a thermogravimetric analysis graph when we're calculating the weight loss. So we have a material that's well characterized, injected into the mice. So you can see that there's enhancement in the water dispersibility of the material upon functionalization and conjugation to the peptide. And because size matter, we have really to see the dimensions of the two components. So these are the CNTs of about 20 to 30 nanometer in width. And this is how thick an endothelial cell, brain endothelial cell. So they're much smaller than conventional cells. So it's quite a challenge to bring this across. So with ultrastructural imaging, so this is the first direct imaging method we utilized, and we want to see how the CNT is actually crossing here, or do they cross in the first place. So through TM, we can actually, this is the TM image showing the intact tight junction. So it's a real model. You have the CNTs interacting, transcytosing, and more likely here it's a macropinocytosis, and this is within 4 hours. So after 24 hours, you see more internalization of the CNTs in a vesicular structure, and within 48 hours, you see the exiting of the CNTs across the blood-brain barrier cell. So STEM images shows you exactly the electron-dense membrane here due to osmocation, and you can see that actually the cells are healthy, intact mitochondria, and with time they're still within the cell, and then they're clearly exiting, keeping the cell happy and healthy across the membrane. So at 4 degrees, the process is completely inhibited. There is still association, but something blocks the CNTs from internalizing. And you can also see them in other type of non-endocytic cells, such as the astrocytes. So with gamma counting, you can see that angio-BEP conjugation double almost the amount of the CNT internalization, and actually even up to 72 hours, you can see this improvement, which is, I would say, twofold improvement. So with Spec CT imaging, we take the mice, the healthy mice, inject them intravenously with a dose of the CNT, and it's important that we perfuse the animal to remove any amount in the blood with heparinized saline before killing. Before killing, we do the repetitive imaging. So you can see here with the non-targeted material, actually here we have to adjust the min-max, so to show you the difference, but even with the non-targeted, we still see uptake within the brain. And this is much more improved when you have the angio-BEP conjugation. So we could conclude that active targeting in this case has improved the passive targeting, the already intrinsic ability of the CNTs to cross the BBB. And you have approximately, so this is per gram of tissue, you have about 2% per gram tissue in case of the targeted CNTs versus about half in case of the non-targeted material. And we could notice in all cases you have very short blood circulation time. So there's no correlation between the blood circulation time and the brain uptake. So looking at whereabout in the brain, it matters. It's not about which organ, but actually whereabout in that organ, because if I want to treat a disease that's localized in the midbrain region, I want to know where they go in the brain. So we take the brain sections, and by autoradiography we leave them to decay for a couple of days, and then you can see that midbrain regions seem to be the target of the CNT, which is quite promising because there are many diseases that's happening in that region. So we know where they are in the brain. We know they cross the brain, but actually, are they reaching the parenchyma or they're stuck in the capillaries? So the capillary depletion methods allows you to take the capillaries from the brain parenchyma. Our target is the brain parenchyma. And these are the doses versus time, where you can see that the targeted CNTs show higher uptake than the non-targeted, but actually this is a total brain. If I look at the capillaries, there's no such a statistically significant difference between the two material. But if you look at the brain parenchyma in particular, you can see that the angio-BEP conjugation actually improves the shuttling into the brain parenchyma. So we can conclude that you help the BBB crossing, but you also have more of it in the parenchyma region with the angio-BEP conjugation. So this is the blood to brain parenchyma, and you can see our ratio is above 0.1, which means more than 10% of the amount in the blood is actually going to the brain. Most CNS active drugs have about this value, so we have a potential that this could be used as potentially CNS delivery system. So with Raman imaging, we want to actually look at the direct imaging of the CNTs. So CNT have characteristic DG signals, and unfortunately we couldn't see any signals in the whole brain, but we weren't surprised because we know that the amount in the capillaries were still greater, and you can confirm their presence. So we actually tracking the CNTs themselves. The multiphoton imaging properties of the CNTs were also utilized in the whole brain sections, and these are cross sections of the brain. The red signal are showing the brain capillaries, while the broad emission of the CNT indicate, showing here in white, that actually the CNTs are present in outside the brain capillaries as well. These are just 3D images showing the CNTs in the brain. The TM shows you whereabouts, how they interact with the brain capillaries, and that they actually also present in the whole section. So because our aim was to target the glioma here, so we used the GL261 brain tumors, and interestingly, after 24 hours, we can see the clearance of the CNTs, whether they are targeted or non-targeted. But in glioma brain, actually, you have higher uptake of the non-targeted one, and more significantly higher with the non-targeted, but also more with the targeted CNTs, showing also promise in brain targeting. So I hope I convince you that we're able to track the CNTs by multimodal imaging techniques, either by indirectly tracking them with a radiation and benefiting from the different resolutions of nuclear imaging, and also by direct imaging of the CNTs relying on their photoluminescence and Raman properties, and obviously the TM is very helpful here. I like to thank the students and the postdocs who directly worked on this project, my collaborators on the different aspects here, and also the funder and my group, and you for listening.
Thank you very much. Questions? Two over there. Yes?
We saw excellent penetration of these carbon nanotubes through the blood-brain barrier. But my question is how these non-water, non-lipid soluble, what will be the fate of these materials in the brain later? Cannot cause inflammation or some other changes later, because how it will be degradable.
So after we check the cytokines level after 24 hours, and you have a slight increase in the cytokines, but it subsidizes after 7 days. And from the SPECT imaging, although it's quite, we are surprised that actually we have complete clearance after 24 hours, so there must be a pathway of clearing them. If you do a local injection, obviously they stay longer. So it's probably worth looking at clearance mechanism from the brain versus following systemic administration. But these are short and functionalized, so they're really more like nanoparticles, I would think of them, rather than big sticky needles that persist in the brain. But the quick clearance is worth investing later and finding out how actually do they clear. It's something that we haven't answered yet.
Yes.
So, Khuloud, thanks for a great talk. We're hearing more and more today about local administration of nanoparticles, not only intravenous administration. Do you see an advantage of inserting a large amount of nanoparticles to a specific disease site in the brain and then have them distribute from there? Would that be advantageous, do you think, over an intravenous administration?
I mean, I've showed in the beginning the therapy study was done with local administration, okay, but the rest of the study is with systemic administration. Probably after we answer all these questions about the toxicity elimination, if we know if my disease is localized, then we can think of the local administration if it's a brain glioma, for example. But for something that's CNS degenerative diseases, you cannot really do local administration. So probably we need both, depends on what we want to treat.
Okay, two more, brief question, brief answer, please. Klaus Michael.
Yeah, Klaus Michael, Saarland University, Germany. Nice talk. The advantage that you showed of the brain penetration was due to some biospecific form of surface functionalization of these nanoparticles. Question is that an effect that is unique only for nanorods, for carbon nanotubes, or would you see the same also by using other type of nanocarriers?
Mm-hmm. So I think even without the targeting, we have intrinsic ability. So my personal belief so far is the shape matters. And
do you have a control for that?
Yes, so the control is the one without the peptide, where
yes,
we
that's
still have...
that's the carbon nanotube without the biospecific functionalization. But I'm wondering to what extent is there a possible synergy between that surface modification and the shape and other features that is unique for nanorods, tubes, or could it also be translated to spherical polymeric nanocarriers, to liposomes and things like that, so that you really need, with all the novelty and potential risks, according to the question, that it is worthwhile pursuing with the carbon nanotube, or could you just use this surface modification, but instead use the established safe other type of carrier?
I think that would be the sensible approach, is to learn from the shape here and you replace it with a safe biocompatible material. And if someone here is interested in making this type of material, we could perform these studies as well.
David?
Thank you for the interesting presentation. First, one small note that it appears that they are functioning as aggregates or conglomerates, and probably the dozens nanometer scale size range is not very relevant to the structures that we saw in the majority of the slides. But this is just a short note. The real question is, do you know what is the corona that is formed on the surface of these tubes, whether it's dependent on the density of your coating with specific peptide, and whether the peptide stays actually on the surface, or it's covered, or it's cleared away after a while in the body?
What we know is the one with the peptide differ from the one without the peptide, but we have not really studied the protein corona, what type of protein adsorption happens on each of them, which could be, which is an important aspect to study. In terms of aggregation, I wonder whether the material is aggregated and they enter as aggregates, or the cell recognize them and internalize them as aggregates. So we see them as aggregates inside the cells, but how the cell probes them and internalize them is something we don't know.
Okay, thank you again, Khuloud. Nice lecture, nice discussion.